what I want to try and give you a little bit of is the perspective of how we go about research in this area, which is a really challenging thing to study as a chemist. Maybe a little bit about why we do it as well. There'll be a lot of chemistry in this because I'm a chemist and I can only talk to you about chemistry. And I apologize if you don't like chemistry. I know lots of people don't like organic chemistry. Bear with me. I will try and keep jargon to a minimum. If there is anything that doesn't make sense, especially in terminology, things like that I say, feel free to stop me. I might lapse into organic chemistry speech by accident. But there will be a lot of organic chemistry in here, but I'll try and give you a light touch on it from the way in which I describe it. But so I came to this field as a true synthetic chemist. Okay, why I'm in this field, I think, is important to say in the context of a lecture like this. I came to this field as someone who worked for AstraZeneca, which is a big pharmaceutical company, and I didn't like my job. I wanted to do something different, and so I went to look for a PhD, and this is when I found this field. I wanted to do chemistry still. I liked doing chemistry. I just didn't want to work in the pharmaceutical industry. I wanted something more fundamental, and this seemed like the perfect opportunity, this field, where chemistry could play a part in understanding something truly fundamental about nature. And it just struck me as amazing that we could use organic chemistry instead of to try and make money out of drugs and out of people. It's a great thing to do, but instead of doing this, instead of focusing on targets of we want to make a molecule that will have a specific function in the context of biology, we could use chemistry to try and understand where biology came from. And when I thought about that in the context of what I knew about the history of life, the history of Earth, which is still pretty rudimentary, it was even more rudimentary then, it's just how immensely long ago the time frame was that we would be applying chemistry to. We wanted to understand something that happened 4 billion years ago. And to me, as someone coming from outside this field, that was an inordinately long time ago. I thought of dinosaurs in here, in this little gap here, as being incredibly long time ago. But that's nothing in relation to the question we're asking, the distance in time, and the fact that we could apply chemistry to try and solve some of this problem, obviously not all of this problem, it was just fascinating to me. And as I became more deeply embedded, what it struck me is that chemistry has a real benefit over lots of the strategies that we would have usually applied to understanding the history of life. That it's not tied to any record. So we heard lots about life and phylogenetics in the previous lecture, which I think is wonderful, is interesting, but we're tied to what we have. We look at the phylogenetic record, we compare the organisms we have, and they're our data set. We can look at fossil records, and we have the data set that we have. We're restricted by what nature provides us in that context. And organic chemistry is a little bit different. And I recognize the criticisms that we get working in this field. I get these criticisms, right? People say them to me. The criticism that's most often presented to us and the way we do our science is that it is organic chemistry. We get this backhanded compliment. It's beautiful organic chemistry, but is it relevant to the geology? Is it relevant to the biology? Maybe, maybe not. But that's one of the strengths of organic chemistry in this field. It doesn't have to be. We can still learn by asking fundamental questions about how molecules react, how molecules can assemble, how they behave, under what environments they behave differently. So the fact that we can do anything we want, we can generate our own data sets, is a massive strength here. And to restrict ourselves to something that is, in everyone's opinion, exactly how the early Earth looked or how early biology looked would be doing a disservice to this field as organic chemists, because we can provide so much more than that. Where that fits within the greater scheme of things is to be figured out further down the road, in my opinion. So that's my sales pitch for organic chemistry in this area. and the area as I see it. And I'm not the first person to think about organic chemistry in this context. We've heard phylogenetic trees already. We've heard the concept of looking at perspective. I'll reference Alfred Russel Wallace here as well. I think he's an amazing story. If you don't know his story alongside Darwin's, you should read it. It's just fantastic, even just from his personal adventure throughout the world to find out the things he found out. But focusing more on what this great man said and what this great man thought, I find it amazing that Darwin, so long ago, 150 years ago, could ask the question that I'm interested in. He can formulate the same question and it's not been answered in all that time, despite that it sits there as an obvious question for science to answer. So whether you like his mixture of random things that he put together to build cells or not, the question remains, how do we build a compound, a protein as he called it, something that is in a cell, something that's essential to a cell, such that evolution can begin? What predates evolution? This is an obvious thing to anyone who sees evolution, that it is a hereditary process. So it must have a mother, a daughter, a mother, a daughter. What comes before that? That can't go on forever. Do you think this thing spontaneously assembled, fully formed at the origin of the universe? I would guarantee you don't if you're sat in this room. Some people, perhaps panspermia, do. But again, we've heard that's not scientific. We're not asking a question that we can interrogate with evidence-based answers by undertaking empirical experiments in whatever field we come at this from. So this is the question that Darwin formulated 150 years ago. It's exactly the same question that drives me. It's the question that everyone in my lab wants to address. How do we get at the point where we can take chemical reactions and build them into something sufficiently complex, sufficiently integrated, that what we now know as life can evolve from that point? This is a really difficult question because we don't know what that thing is. We don't know what it looks like at the origin of life. We have nothing to go on other than our best guesses. We know what life looks like now. We have some guesses. They're kind of weak in my understanding of it, of what the early Earth looked like, and they tend to be global references to the early Earth. Do we know life didn't form in a niche environment? I don't think so. So what we have to go on at the beginning of our journey is very little. We just know we need to work our way back towards that, and so it's a very, very complex question to answer, and there are lots of ways to try and answer this problem. I am not saying our way is the only way, and it shouldn't be the only way. There has been a lot of approaches to the origin of life, and they are ongoing, to take a geochemical perspective on this problem. And I appreciate this angle. It is not my angle. They rely on picking a scenario, a type of chemistry, a place where you think the origin of life occurred, and then investigating that chemistry. You tie yourself to this scenario. And this is what I'm saying organic chemistry in this field doesn't need to do. We don't need to assume life started by lightning discharge or by submarine vents or in a meteorite impact. We don't need to tie ourselves to any of this. We could ask fundamental questions about how to build molecules without having that inference, without having to have faith in any one of these systems. And that is the approach we take. We try and extract chemistry away from geochemistry to look at how molecules behave, how they're made. And we do this because that's what I'm comfortable doing. Right? I'm going to admit that to you right now. Right? None of these other approaches I'm describing are wrong. They just take a different skill set than the skill set I have. The skill set I have is understanding reactions, understanding how organic chemistry works, and those are the skills I want to bring to bear on the problem without being tied to anything else. I want the full range of my abilities to be allowed to be used to address this problem. And I you can correct me if I'm wrong. I'm very happy if you can tell me where the scenario is and the experiments we should do. But I feel that the geochemistry we know from the early Earth is not sufficiently well defined that we can pinpoint where and how life yet started. So what do we do about that? Well, we can look at life, we can look at what life looks like, but again, we end up with these thresholds of what we can learn from biology. How far can we go back? Can we push all the way back to the origin of life? We come to the point where we can't add much more, I skipped lots of slides there, information on this. We get to kind of speciation thresholds. We can't, with the information we have, see beyond a certain point. And so what do we do about that? Well, the standard thing we do as organic chemists in this field is we try to make models. This is a great thing to do, to make a model so that you can simplify the problem and you can then address some part of the problem and then come back to the whole. Lots of science works this way. Almost all good science works this way. But we should never forget that our model is a model. We should never think that we have something special in our model because we made it a model. Just because we're studying that part of the system doesn't make it special. Okay, we have to prove it's special. Our interest in it is not relevant to the problem. That is just probably what our skill set is again. Okay, we have a certain skill set, this defines what we study, therefore we interpret importance in that. Okay. But let's look at what people have commonly done as prebiotic chemists, people trying to bring chemistry to bear on this problem. They look at the cell, we simplify what's going on in the cell, we think about it in the context of information, in the context of trapping energy within a membrane, and that we may need catalysis to perform some of the functions that go on in this process. And there are all these things, all of them are integrated in biology. The easiest thing for us to do from the perspective of studying, trying to manipulate this system, to learn about this system, is to start separating them out. Start inferring that we can study each of these components, and I've suggested 4 here, you could probably suggest more, and we can look at them. I suggest these 4 because these 4 have dominated this field. Okay, if you joined this field when I joined this field 15 years ago, the field was completely polarized or quadripolarized into 4 camps, where if you sat in one of these camps, you knew what the origin of life was. You knew where it lay. You knew in which molecules it was in. So you could sit in the Darwinian evolution camp because you knew you needed Darwinism for life to propagate. You need information to pass on from one cell to another cell. Or you could sit in a metabolism camp and say you need energy in the cell. You have to accrue energy to build the things to make any of these run, to make these work. Or you could sit over here in the catalysis camp and think that you need to catalyze reactions. Lots of biology's intrinsic reactions run really slowly without catalysis, without proteins. To move the catalysis to help us get to equilibrium, the rates of these reactions are so slow they become unreasonable on the time frames that a cell exists. Or you could sit out here in the compartmentalization camp and say, well, if we don't segregate the cell from everything else, we don't have life, right? We can't have life unless we define what's in life and what's outside life. If we don't have an environment, we don't have life. Without this separation, we don't have anything. Okay? And each one of these holds merit, right? But the question is, do each one of these hold merit on their own? Is it still true that you can run Darwinian evolution without an energy supply? Is it true that the catalysis is valuable without something to program it, that can then feed back into the replication of the catalysis that you have? And everyone picked their favorite and went with it, right? So if you were a Darwinian person, you thought nucleic acids came first. These were the be-all and end-all. If you had them, everything else didn't matter. And you can go through them all, right? There were camps for each of these. And people who sat in each one of these camps, therefore, came to the conclusion that the first molecules were metabolic. The first molecules were informational. The first molecules were membrane. Okay, they extracted all the reason for these molecules having useful function. So a membrane without bits to keep inside, what do you want it for? It's a bag without contents. What are you carrying around? Nothing. The metabolism without something to build with them, what are you doing? You're just doing chemical reactions. You're not sustaining a cell. So none of these actually are meaningful on their own. You can extract out each of the individual components and study them. That makes the task of studying each of the individual components easier. But by extracting them out so you can study them, you shouldn't infer from that that there is anything special about the one that you extracted out from the whole. Life is a product of the whole and all of these, and ultimately we need to build a system that has all of these things, that all come at the point of having a living cell. Is it meaningful to say one of these came first? I think not. Okay? I don't think that is relevant, meaningful, or useful. Is it helpful to take one of those subsystems and say we're going to investigate that one, see for how we can push it, what we can learn about it? Yeah, that's helpful. That lets us do empirical experiments. We can get data from that. But that doesn't infer importance for the origin or the order in which these came. Okay, so just a little bit of warning, a little bit of care, and if you want to see pictures of why this is true and where we look at these things in biology, well, here they are. So we've got DNA, protein syntheses, we've got our membranes, here's metabolism running across the membrane. There is not one of these pictures in which we're considering the biological context of these four scenario elements, whatever you want to call them, where the individual part is operating in isolation. They are all integrated and they are only useful in terms of building a cell when they're running together. Okay, so this has been our premise. As long as I've run a lab, as long as I've worked in this field, before I ran a lab was to think about the whole, okay, not to extract out any one part and give it importance, but to recognize that we can learn by making those leaps of our minds, our invention. We think about one of those systems in isolation, but recognizing that it's got to go back in. Once we've learned what we learn about nucleotides, we have to put it back in to the complete system for it to be meaningful. And that then can infer how we move forwards, what we learn about next. And this is a reiteration. This is how our lab runs, right? This is what we think about on a daily basis. Our goal is to build something that could act like life, something that could realistically evolve to become life. Okay, not an imaginary concept, an artificial thought experiment of what life could be. We want to make something that is actually on a realistic evolutionary trajectory to what we see here, now, today. Right? We want to do that because we know that works. Right? The evidence we have is life on Earth. The evidence we have suggests that life can get started on a planet, and life can get started in such a way that it evolves to be the life we see now on this planet, which we've got one version. So therefore it makes sense that we would aim for something that is on an evolutionary trajectory to what we know. Anything else is a very cool experiment. It's very interesting, but we don't know it will work. Okay? We don't know that we're going to get to a point where we can actually establish another form of life that works in a different way than the one we find here. So this feels like you're on a winner. Feels like there is some trajectory there that you should be able to get from chemical reactions to a cell like you or I, or all of the other cells that are on the planet today. But the questions we need to ask ourselves is, do we? And I've kind of told this that you do, but you should continue to ask the question, do you, right? Do you need to separate these systems? Okay? Can you have one of these systems operating in isolation? Maybe I'm wrong. Maybe you don't need to take a holistic approach. I think you do. You certainly can challenge that view, right? But I don't think it's going to be productive for you for lots of reasons. But if we have these inherent chemistries, and we have these inherent differences in how we have different molecules, so we've got genomic molecules, and if we look at the ones we have today, they're DNA and RNA, right? They're built from phosphodiesters, they have this base pairing, they stack, they use specific chemical interactions that we can completely understand. So phosphodiester ligations with stacked base pairs that allow us to separate those structures from bulk aqueous solution and transfer information from one side of a duplex to another side of a duplex. We can understand what our catalytic molecules, most of them are built of peptides. They're different bonds, they're different structures. They are different. Same with the lipids, right? We can understand all of these. So then it becomes important to ask, well, do these systems, do the chemistries that make all of these things, are they orthogonal? Can they operate together? Can they operate in such a way that they don't cancel each other out? Again, I think the answer will be yes. Biology's not chosen to make peptide bonds for catalysis and peptide bonds for information. Why not? Could be easier, right? You only need one form of bond activation if you're going to do that. There's got to be a good reason. Why pair up two completely different sets of molecules to pass information and catalysis from generation to generation? Perhaps they are orthogonal, perhaps they can be made together, and perhaps by pairing them lets us separate information and catalysis. There is virtue in doing this. So can we use these systems? Can we define these systems? Can we think about what they are chemically to understand why they were built together, why they were selected, or why they were presented? It could be a combination of both selection and what is presented to the initial cells as they form. And this ultimately becomes the question of, can we self-assemble this? Can we put it together? Can this system, can these chemicals be built together, respond to each other's presence in such a way that they become functionally useful? And by functionally useful, we mean can evolve, right? Can replicate, can pass information, can extract information and catalysis and energy from the environment. And I could add environment on here, like how does this fit within an environment? But I'm much less capable of doing that because I'm not a geologist. So I'm going to leave that, I'm going to sideline that and leave that for somebody else to do. Figure out how this fits into a geochemical scenario. I'm ill-equipped to do that. But then once we've done this, we've set up a whole set of questions. Very, very broad scope questions. We need to think about what we're going to do in the lab to define actual experiments that we can do. Okay, things that we can actually test in the lab. Okay, with no faith, we don't have faith that this is got to be in this way, it's got to be done like that, this has to be that. Just how can we ask general questions that let us set up an experiment and get an answer, yes or no, that then we can feed back through a cycle of repeating another experiment to test another principle. Okay, and to do this, we have to think about the systems and the parts of the system. And this is where it comes to us that we then have to start thinking, which one are we going to look at? And there is no importance. It doesn't have to be any one. But you have to define what question you're going to ask. What is the answer that you want or expect or maybe hope for? What are the other answers you might get? And what do those different answers tell you about the assumption you initially made so that you can go back around the process of discovery? Okay, so we're going to experimentally evaluate all of these subsystems, and as I said, we could define more, but we're going to look at the genome. Can we build these bits and how do they function? We're going to look at the type of molecules and types of catalysis that we can do. We're going to look at what it means to be. This is the one I find the most difficult, and if anyone wants to have input with me here, feel free to shout things out. But what is it? What is meaningfully metabolism before you have life? What does it mean to be metabolism without a cell to sustain? Okay, metabolism, as dictionary defined, are the reactions that support a living cell. So what does it mean to take metabolism away from the cell? They're no longer the reactions that support a cell, they're just reactions. Okay? So without context, without the cell, what is metabolism? I still don't fully understand this. But it's an interesting concept. So we can separate it out, and we can certainly think about the types of molecules in biology that we would associate with how biology extracts energy from the environment, how it extracts carbon, nitrogen, phosphorus, etc. from the environment and couples it into these processes that let it build genomes, catalysis, and membranes. Is it metabolism without these? I don't know. I feel like not, but again, this is a semantic argument, but it really doesn't mean very much to make molecules that have energy unless you have a purpose, a use for them. And then once we define these four things, how do we make sure that we're not just running 4 or more separate projects? Right? How do we define that we're not just doing RNA chemistry? How do we find that we're not just doing peptide chemistry? Because people world over do peptide chemistry, right? Try and understand peptide chemistry. How do we ensure that we're still saying something meaningful about life as a holistic system? We have to keep comparing them. We have to keep contrasting them. We have to keep understanding if there is interplay between the different systems. We have to ask ourselves, are intermediates en route to these the same? Are intermediates en route to these different but formed by the same process? If we have those different intermediates en route to these things, are they compatible with each other? So not only do we have to make a system where our genome is compatible with our catalysis, which is compatible with our metabolism and our envelope that encloses all of this, if we're going to use other compounds to get from the very simplest chemicals, ultimately if you want to push it that far back, we don't, but right the way back to atoms, right? We have to understand that the intermediates are also compatible. That if we have a different intermediate for catalysis, whatever we make our peptides from, different intermediate for our nucleotides, that those don't self-annihilate, that those are self-consistent within one system. And if they're not, we then have to address that. Do we realistically believe that we can have part of our system set up in pool A and part of our system set up in pool B and then bring these together? If we do, we then have even more complexity to the questions we're asking. Why is pool A useful and pool B useful before we mix them? Okay. So we've got to keep asking ourselves about these as a total system before we can say anything useful about any one of the individual components. And then once we do this, we can start to push the boundaries of what we can actually address. And this is where we need help. Right? This is where chemistry stops. I can iterate these ideas and go around these cycles forever. Certainly keep a group going like this until I retire. But again, to make it meaningful, we then need input. What do these chemistries say about geochemistry? So rather than having the geochemist tell you this is the environment where you should be, I'm happy to have that. We're happy to try some of these things. What does the chemistry say about the geology? So let's assume we can have many different geologies on a diverse planet. I don't see why not. There's plenty on the Earth as it exists now. What do the different chemistries say about the likely inference for where we would have life? So we heard lots about phosphate this morning. If phosphate truly is an integral part of this system, then the chemistry is telling us something about the geology. Right? The chemistry is telling us we want to go looking for geochemical scenarios where there is phosphate, if phosphate is truly an integral part of this system. Once we have made that leap, we don't make that leap to pat ourselves on the back and say, hi, we found a geology friend. We can then tell each other we're right. We then have to ask, what does that tell us about asking new questions? What does that inform us? Knowing there may be a geochemically relevant environment, what does that do to inform future experiments? And the way we do that is then you would look at what does that environment suggest? And what does that environment mean in terms of chemical consequences for reiterating this process? If phosphates come with sulfides, what do sulfides do for this system, for example? So each one of these has to be set up in such a way that we can keep learning, we can keep in mind the target, which is the whole cell, and that we can keep going back around this process of discovery. There is an end to this process until we have the almost impossible task of building a complete, functioning, living cell. We're probably not going to get to that point. So there is no point where this set of questions we ask ourselves in the chemistry lab will be done, will be finished. It'll end. It can't end because life keeps going, right? The chemistry that underpins life keeps going, keeps evolving, keeps changing. As the environment changes, I would guarantee, and there's someone sat down here who's better to discuss this one with me, but as the environment changes, perhaps because biology is influencing its environment, the properties of these molecules will also change in response. So this is an endless question we can keep asking. As we change the environment with biology, we will change the way biology operates, and I'm sure we can see that in the phylogenetic record. As we have different molecules that provide us our energy, we will change the way metabolism runs. As we have different environments that life seeks to push out into, the membrane will change in response to what is fittest for that environment. So these are endless questions, which is good when you're an academic, because you don't want your career to end, not until you want to retire. So that's good. But so now I'm going to introduce some. This is, I think, my first organic chemistry. Okay, here it is. Hopefully these are all molecules you're familiar with. This is also a concept that was brought up this morning, but now I'm looking at it from a completely organic chemist perspective. So I have carbon molecules up here, okay, and I've picked and chosen some that are relevant to life, okay, and on the underneath I have an array of compounds that are abiotic, right? Things that you could definitely ascribe to being provided by a planet, right? Without input of biology. So we can all agree, I hope, that these ones are biological in some sense, and these ones are potentially inorganic, if that's the right word. They're potentially geochemical. They're things that we could build on to make life and have them in an environment. So what strikes me straight away about these is the parameters within which life works, right? And the parameters then, as I look at what people discuss in terms of geochemistry, where the geochemistry operates. Okay, so I hear a lot about CO2 from geochemists. Okay, that's hard to work with as a chemist, but okay, there's plenty of it in the environment. I hear a lot about methane from geochemists. This is also really hard to work with as a chemist, and it's also not where biology works. So biology works almost exclusively between plus and minus 2. Okay, all the molecules built in cells are in there. Okay, so we then, as chemists, may want to ask ourselves what's special about these oxidation states. Why is it that we can build complex molecules that are then functional out of these molecules rather than the ones on the ends? Okay, there are some plus 4s in biology, but these are the ones that really grab our attention as the molecules we want to make. And we're thinking about this system of metabolism, if you want. I've got some metabolic molecules. I've got some that may predate the molecules of information, and some of peptides, and here's some lipids out here. Why is it that the biological molecules all fall in here? What is it about them? Why should we address these? What does it tell us about the chemistry that we should do to build a biological system that it falls into this gap in the middle? Why do we have a mix of oxidation states? Well, there's lots of beautiful chemistry, and lipids, I really like lipids, and they're a beautiful example, and we do the least chemistry on lipids, right? Because I think they're so well understood that I don't really need to do very much. But they're wonderful molecules, right? They do have properties that make them quite special. But so many molecules have those same properties. So if we have a long tail and a polar head group, then we can assemble these types of molecules into their amphiphiles, is what we would call them, because they've got two different natures, polar side, apolar side. They assemble, they self-assemble, they build themselves into structures. So it very quickly becomes apparent that if you take very simple compounds, in terms of the complexity of how we build the different bonds, the difference in the bonds that are there, very just simple linear fatty acids will self-assemble into structures that look straight away interesting. Okay, we can take a molecule like this, if we take it at about pH 8, which is its assembled pKa, so we can learn lots and we can understand all kinds of things about the chemistry. So it's a fatty acid. the dissociation constant for it means that it will be half deprotonated in solution at about pH 4 to 5, depends on the exact acid you have. But these will aggregate, they will assemble into structures that start to look, if you kind of squint at them, like a membrane. If you put them at pH 8, and this is because once you self-assemble them, the pKa changes. Okay? So by knowing how this molecule behaves, if I took it at pH 4, which is its pKa, where it's half protonated, half deprotonated, it won't assemble like this. If I take it near neutral pH, near 8 ideal, but 7, 8, it will start to self-assemble because it's half protonated in the assembly. As I try and push these negative charges together, they don't want to be together. They repel. So it changes the pKa. It changes how willing this acid is to give up its proton to become the anion. And in using this, we can assemble structures. And this has been known forever. As long as people have done organic chemistry, people have known this way back into the 1900s. And as long as people have been studying the origin of life, people have said this is important. Okay, they have this function. But is this function useful and productive? Well, if I take one of these amphiphiles and I want to study it, okay, and this isn't our experiment, this is an experiment done by a colleague, an ex-colleague of mine in the Shostak lab, Ting Zhu, but I find it interesting when we're thinking holistically, when we're thinking about the system, when we're thinking about what's in the system and how we study the system as chemists, and how we make our lives easy for ourselves by making things simple. So if I make, or if Ting makes, I wouldn't like to do it, but if Ting makes, if Ting makes a unilamellar vesicle, right? So it looks a bit like a cell, right? I've got an inside, I've got an outside, I've got one cell membrane. It's composed of fatty acids, could be phospholipids, composed of whatever you want. And if I add to this more material, I can see growth, right? This still sounds biological, right? This sounds really cool. I take something that looks like a cell as I just look at it. It doesn't function in any way like a cell, but it looks like it. And we infer a lot from looks as human beings. So we like the look of this. I can see it under a microscope. I look at a cell, I look at one of these, they look, oh yeah, good. They're the same thing, right? I add more material. I don't worry about where I'm getting it from, chemical synthesis wise, but I add more material. So the same substrate, okay, this same substrate that's built my unilamellar vesicle, I add more of it and it grows. But this growth is so dull, right? I have a sphere and I grow a sphere, it grows symmetrically. I get nothing else. I take a vesicle, I add more material and I get a bigger vesicle. And you can study this, people did, for years and years and years and years and years and years and years, but you don't learn any more, right? I get this thing and it grows. But so then you have to think, and this was, I'm not sure whether it was laziness on Ting's part or it was actual real thought about what are we going to do with this? You have to think, where did this come from? Right? What did we do to get this to study it? The way you get this is you take your lipid molecules, you shake them in water, right, and then you let them do their thing. They form stuff. You don't really know what it is. You can't really study it because they're all different. And then you extrude it backwards and forwards and you push it through sieves, little membranes that force it to form one specific size. And you make it into something that is sanitized. Once you have all of them the same, single layers, all the same size, you can look at them all under a microscope and it's meaningful to the eye. You can see them, you can study them, you can do science with them. And you can learn about, you know, the on rates, the off rates, all rather dull chemical equilibria things that you can investigate in a system like this. But that's it. Right? So you simplified the system to the point where you could study it ad infinitum. But what you learn is that you can grow something small into something big, and it didn't do very much more interesting. What Ting did, and this was a brilliant step, difficult for him because he had to make a new way of making these so that he could look at them under a microscope, but he extruded them in a different way so that he didn't take away what you initially get. So he made their sizes similar, but he didn't make them unilamellar. So when you initially extrude these molecules, the thing you get when you just shake amphiphiles in water is layers upon layers upon layers of membrane. Okay, and they behave completely differently than the unilamellar versions. So this is more like, not exactly like, right? He's still changed the system a little bit so he can study it, but it's much more like what he gets. And it has a great deal more heterogeneity. So we have layer upon layer of amphiphile bilayers. And now when you add material, it behaves very differently. So it can't grow spherically anymore. We don't have a uniform sphere. We have an outside and inside, and the outside and the inside kinetically have to respond differently to the material we add. So as we add material, it has to first enter the first layer, and then it has to go through a process of moving through that bilayer, which it doesn't want to do. It wants to insert in that bilayer and stay there. Then it has to leave the bilayer, go through a bit of water, and go back into another bilayer. This gives you a kinetic barrier to growth inside relative to outside, and every time you have another layer, it gets worse and worse and worse, slower and slower and slower, and this means that you can't grow spherically anymore. Mathematically, it means the outside has to grow faster than the inside, and therefore you have to start adopting a different shape. So these don't grow spherically. They don't really look like this. I just drawn two. Well, it's actually stolen from Tings. He's drawn two to keep it looking simple, but there's lots of layers in here. They have to elongate. They have to grow in different shapes. So they grow in filaments. And then once they grow in filaments, filaments are no longer stable, unlike spheres. So they bud. They form little daughter versions of themselves, and then they can grow and go through the same process. So by keeping some level of heterogeneity in the system, we get back function. So this is and I can show you this process. I hope this works. How do I? So this is Ting's video. It's in his, it's in his paper. If you want to see the real video, this is the real video. If you want to see it in its native environment. So here's one of those blocks of micelles that are all laid on top of each other. Material is added. It adds and it grows in a filamentous way. And then this is kind of hard to see, but these filaments then in solution, you pass air over the solution. And as air passes over the solution, it causes instabilities. And these instabilities then cause division. And if you can see why these are coloured, is they have dye in them. Okay, so there's something bright, something that the camera can see, can detect. This dye is all kept inside. Okay, the background stays dark, inside stays light. So as we go through this process of growth, now we add division and we maintain encapsulation. So really simple principles of chemistry, really simple principles of physics about how we grow and how we cause instability such that we can get division that allow us to get cell-like properties. But there is no way these are cells. These are not alive, right? We've still got to put the rest of the stuff in there to understand how this becomes useful in the context of moving forwards in the origin of life. So we have a bag. What do we put into it? We have a mechanism to make these structures, simple self-assembly. This is one of the questions I asked. Can this simple system self-assemble? Well, certain parts of it obviously can. We can divide it. Oops, going backwards and forwards. We can divide it. We can keep the things inside. We've got inside and outside. This is some way towards building a model to understand where our first cells might have come from. Our lipids, well, Ting's lipids, amphiphiles, look in some way like they might relate to some of, not all of, but some of the biological lipids we use today. You could see how there might be an evolutionary trajectory from there to here. So this kind of fits. It's all going very well. But then we move to the inside, and this is where it becomes chemically challenging. Now we've got to think about how we make these molecules. Where do they come from? They're no longer simple. Okay, so once we move inside the cell, Then we find other things that are uniform about cells, right? We find the ways in which we move information in cells are complicated, really complicated in a chemical sense. But they're uniform to all life, and they're kind of, you can kind of see how some of the processes of self-assembly might work with these, right? You can make DNA molecules, single strand, you can present the complement, and these things, coalesce. They find each other in solution. They will come together. Same true of RNA. Peptides, they're more complicated. These are quite difficult transitions. But so, lots of people have tried, because particularly this transition here, right? I can make a single strand piece of DNA or an RNA, right? And I can present its complement, and both of those will come together. I can present a DNA with an RNA complement, they'll come together too. You can feel chemically why this system works, right? So lots of people have tried to take this bit off, as though this is later invention. I think that's a mistake. I think this is what's in biology. The clues about how to build this system, not about how the individual components function, but why we might be able to make them, are embedded in all of these species together. Okay, and this is our holistic approach again. Don't just take out one of them and say, that's the answer. We will study that in isolation. Think about each of these and think about them in the context of this as well. Why would these come together? What is it about all of these that has relationships? But we're good scientists. First thing good scientists do is not to define experiments you're going to do, is you go and look at the literature, right? You go and figure out what experiments other people have done. And lots of people have done experiments, particularly on amino acids. So here's a classic. Here's maybe the first, I don't know. Certainly the first anyone talks about. Miller-Urey experiment. Back to one of those geochemical scenarios. Right? We've got an impression of what we think the Earth is like. It will provide. Okay, we're going to take a gas mixture that we like the look of. We're going to pass huge amount of energy across it. We're going to get stuff, and we're going to then see what's in that stuff. And we're going to hope it looks like life. So there are a few things I have to say here, and I think this is a beautiful experiment, don't get me wrong. I think it's massively influential and a very important experiment. But it's not the same as saying we've done this experiment, we've seen a few amino acids, therefore this is how biological amino acids were provided. There is lots of things that we can sweep under the carpet here if we want to just think about this in the context of amino acids. We should also think about the context of other molecules being synthesized alongside it. Are they relevant to our system? Are they not relevant to our system? Have we not hit the nail on the head with this idea because of the chemistry that we've presented isn't exactly like biology? Is there something else? The first solution is not always the best. Right? The second is not always the best either. But if we look at this is Miller doing his stuff. This is his setup of his experiment. We've got water. We heat the water. This gives us vapor. We've got an atmosphere of methane, ammonia, if I remember it rightly, and I don't know what else was in there. Miller-Urey experiments. We passed spark discharge across it. Lots of different things. But this is the first analysis that was done. So the first analysis was done, 4 things show up, maybe more that aren't assigned. 2 of them are biologically relevant peptide type molecules, right? We've got glycine, we've got alanine. We then get beta alanine. Now this is arguable. It's in the cofactor, so it's in CoA, right? But it's not in the genetic code. So maybe it's biological, maybe it's not. Why isn't it in the code? Don't know. And then we get other things that are definitely not in biology, and lots more. And remember, this is just a small subset. Right? They take a huge gamish of molecules. They extract them into really concentrated acid, hydrolyze everything, and then separate out all the compounds and look for the ones they're interested in. And they find some of them. But don't forget about all the other ones. So here's a later version. I'm sure people could keep doing these analyses forever and keep finding molecules. So this is a later version of the same type of experiment where we come back and analyse and see what's in there, and the complexity has gone up. But is this good complexity? Is this interesting complexity? Well, it depends on your perspective. Does it look more like life? Now we took a closer look. Maybe not. So if I colour red, now the components that we don't find in biology, in biological proteins, they get pretty pervasive. Most of these compounds are not biologically relevant compounds. This, to me, is a concern. There are mechanisms, therefore, to make biologically relevant compounds, protein-type molecules, but they're very loosely related to the ones that are actually exploited in biology. Is this selection? Do we just give that process? We're chemists, we're done. Here you go, biologists. Have all of these compounds to play with. It's your responsibility to clean them up. I say responsible. Chemists are good at this, causing environmental problems and then letting somebody else clean them up. But should we therefore look a bit more broadly? Is there another solution? Is there something else? Is there something, even if this is the right solution, is there something missing that's overlaid on this that lets us impose some level of selectivity on top of what we see here? So then we go on, nucleic acids. We can ask all the same types of questions. These are more complicated molecules. We have more bits to put together. Not going to go over them in the same level of detail. We look back in the literature, you find the same types of things. We can take HCN, it's a pentamer. Its pentamer is potentially adenosine. I'm sure anyone who's been in this field. So there should be a synthesis of adenine through the dimer, the trimer, the tetramer, and then on to various molecules, some of which look like biological molecules, some of which don't. You can make lots of nucleotide molecules like this. It's the same story, right? We can do chemistry, it makes some of the molecules we might be interested in, but it doesn't necessarily relate directly to the organic chemistry we find in biology. So is there more to learn? Are there things that we're missing about this system? Sugars. So if we look at the nucleotides, we kind of separated them out again. We've done this thing that we do as chemists where we take one bit and we study that in isolation and look at the other bit in isolation, and we pat ourselves on the back when we find the right bit. But so if we then look at the sugars, sugars are way more difficult. These are really difficult, and everyone thinks there's an answer to this in the literature, but I'm not sure there really is in the thing that they think is the answer. So what people think the answer to sugars is you take formaldehyde, which everyone accepts you can make, and you polymerize it. Formose chemistry. Everyone's heard of this, right? You take formaldehyde, you treat it with a really, really, really strong base, calcium hydroxide, really concentrated, and you get a complex mixture of sugars. Do you? I don't know. Butleroff tasted his in 1862 and said, sweet, sugars, right? He seemed to conclude this. I don't know how clean Butleroff's formaldehyde was in 1860, whatever it was he did. This is an issue. When we look at the chemistry, and this is one of these times when I'm going to force you to look at a bit of chemistry, right? When we look at the chemistry, this is really, really difficult chemistry to do. Okay, what we want to do is we want to take formaldehyde. It's going to hydrate almost exclusively in water. Okay, we're going to take this compound that's an electrophile. It will behave as an electrophile. It will form this diol. This is the compound we're going to play with. We then have to doubly deprotonate this. Treat it with sufficient base that we strip off both of these protons here. The first one's pKa is 10. God knows what the second one is. You can probably take a bit off at 14. And once we've doubly deprotonated this, we have to deprotonate it again. We strip off a third proton, take the trianion, and react this with our electrophile. This is incredibly difficult to do and may, I don't know, or may not work. Maybe it's impurities that make this run. So this is a classic analysis, lots of people have seen this as well, of the products that you get when you do the Formose reaction. But like every other Formose reaction that you will find in the literature post Butleroff, it's not a Formose reaction. He initiates it with glycolaldehyde. This is why everyone who studies the Formose reactions actually studied C3 and C2 chemistry and put those together. Going back to Formose chemistry and making that triple deprotonation, making this solely electrophilic compound behave like a nucleophile is so difficult that people just don't bother. And if you do get to the point where it doubly deprotonates and you're starting to think about taking off this other proton, if you actually look at the chemical literature, all the reactions that people perform to try and do formose mimics, what you'll find is almost all of their material that does reaction doesn't do the chemistry they want, aldol chemistry. It all does this reaction, which is a very well-known reaction in organic chemistry. If we doubly deprotonate a diol, it likes to transfer a hydride to a partner molecule to disproportionate. So we take C2 aldehyde type compounds, we treat them with really strong basic solution, which we think is the solution to making our sugars, and they don't make sugars, they disproportionate and give us carboxylic acids and aldehydes. This is a really common reaction. People loved studying this chemistry. People spent years figuring the exact mechanism of this out. It's really clean, efficient chemistry to do, and it inhibits the sugar formation. Let's put that aside for now, because there are other solutions, and there's always more than one way to skin a cat. I just don't like formose chemistry, and I don't see any good reason why it works. You can correct me if I'm wrong. No one has yet. But let's assume we can do it. Let's assume we can make our sugar. We take that sugar and we're going to try and make these bases attach. And again, this is becoming really difficult. The chemistry to do this is really hard. When we're hitting brick walls as chemists, the thing not to do is to just throw more base at it, not to throw more compound at it, not to heat it hotter. Maybe just take a step back. Is there another solution? Can we think about the breadth of organic chemistry, the types of reactions we can do to push our chemistry in a different direction? And these are just a few quotes from the literature saying about that you can't do this types of chemistry. These are very old quotes, and I worked on this for a long time. I'm not going to go through all this chemistry. Apologies to any chemists, and there you go, non-chemists. But I'm not even here to tell you this is the right way to make nucleic acids, right? There may be a better way of making nucleic acids than this. I don't really mind. I don't need my way to be the better way. What I want you to see is that this is not the only way. Okay? There are other ways. When we think about chemistry, we need to think beyond what is the obvious. So the obvious disconnection here is splitting this bit that's got lots of nitrogen in it away from this bit that's got lots of oxygen in it. And if you went back to the literature post 2002, you would find lots and lots of descriptions about why mixing nitrogen chemistry and oxygen chemistry would be bad, would give you lots of compounds you didn't want. But all these were based on statistical analysis. If I take 3 elements, oxygen, nitrogen, and carbon, I'm going to get more products than if I just take 2. They didn't take into account kinetics, okay, which we always must consider in chemistry. They didn't consider what were the actual reaction pathways that could happen, whether you would siphon material through specific pathways. So the real take-home messages from this paper, and the ones that I want you to take as non-chemists or chemists, is that there is always another way. We can always think around the problem from a different angle. One of the more important points we wanted to make in this paper as well was that if you introduce some of the things that you think and know are important later down the line, you might be surprised, not in a bad way, in that we're going to have this combinatorial explosion of things. We mix nitrogen and oxygen compounds together at the same time. That was meant to be bad news, it wasn't. If we put the phosphate in there that you need at the end, it can be beneficial in many of these steps as well. Try to think about increasing the complexity of your reactions as far as you can, such that you can run them in a way that is meaningful. Okay. But this is not the final solution. This is not the end of this story. Maybe this isn't the right solution. Maybe there's better solutions. I don't know a better solution for pyrimidines yet. Feel free to come up with one. I'm happy for you to do that. It also didn't come without its criticisms. And these criticisms are great, right? These are part of how we develop science. You telling me that there's something wrong with my chemistry is fine, right? This is how we improve the chemistry. This is how we think. Once you've done something, you're so impressed with yourself, right? That you've achieved this thing. It's hard to take a step back and say, How do I improve it? The easiest way to find that is for somebody else to come along and go, yeah, but this thing, it's bad, isn't it? And we had lots of that. And the classic one is this complexity. Okay, so the complexity of sugars that you would expect to go in these reactions. Why are we separating out these two sugars? Okay, so this works beautifully to get really clean chemistry. But these are two sugars, products, we're assuming, of 4-Mohs chemistry at this point in time. So we're going to make it from 1 carbon to 2 carbon to 3 carbon. Why would the 2 carbon be over here and the 3 carbon over there? Maybe that's okay, but the onus is on us to prove it. Right? The onus is on us to explain why. How can that happen? What is the mechanism? We can say, yeah, it could have happened. That's not the same as showing it did, could have done, is not the same as showing a mechanism for how it might have done. So, here's relaying that same problem. If this is our input, then how do we get these two out? And how do we get these two out in such a way that they come in the right order? This one, then this one. So this is not our chemistry. It was developed by my PhD supervisor long after I left his lab. But I think it provides more of those examples of thinking outside the box. Nitrogen, oxygen chemistry. How do we get around these problems? How do we activate this substrate here? How do we make this act like a nucleophile? And their solution is not to do this. When you come across a problem that you can't resolve, a great way of resolving that problem is to just sidestep it, right? Just do something else. Get to the same point. So if you look here, we take formaldehyde and then do 2 reactions and we get glycolaldehyde. At no point do I expect formaldehyde to behave as a nucleophile. No point is this carbon attacking, giving electrons to another carbon. It's always accepting them on this route. So what they do, and they go a bit further back, but what they do to resolve this problem, the 4-Mohs problem, as I outlined it before, is they react formaldehyde with cyanide, a really, really efficient reaction. Rate constants of 10 to the 36. This reaction just goes. Can't stop this reaction. If you take formaldehyde, it's hydrate, whatever you want, treat it with cyanide, it will form its cyanohydrin. So I make they made a carbon-carbon bond here between something that's intrinsically nucleophilic and something that's intrinsically electrophilic, proceeds incredibly cleanly, incredibly efficiently, made our carbon-carbon bond, and then they reduce it to make the compound they want. So they start with C3 and move to C2. And the process of reduction that they use, again, is quite simple. They take a sulfide and they shine light on it. And because sulfide's an anion, it's exposed with light, this has to be UV light, then it will liberate an electron. Lots of compounds do this, lots of anions do this. You don't just have to use sulfide, but this is one of the simpler versions of this chemistry. And we can push it back. So we can take cyanide. We can reduce it with sulfide and light to get formaldehyde. It will then react with cyanide to make to the same 2 reactions to make glycolaldehyde, and then on to glyceraldehyde. And we can make the substrates we want. This chemistry now works. We don't have to beat it, we don't have to force it. It's chemistry that works and works for the principles that we understand as organic chemists. Does it look like how life makes these? I don't know. That depends on your perspective. So if you look at photosynthesis and the way cells make sugars in photosynthesis, well, they attach carbons, CO2 in that context, and then reduce them down. So in terms of adding and then reducing and adding and reducing, it's not a strategy biology hasn't exploited. Does it exploit cyanide? Well, where would biology now get cyanide from? I don't know. I've said already before, how chemistry, metabolism may have changed as the environment changed that it's in. Perhaps early environments presented lots of cyanide. Certainly this is reduced nitrogen, and this was kind of why I was asking the geologist this morning. What happens to your nitrogen in this system? Do you really get Titan-like chemistry where you get lots of nitriles? That would be useful, it would be interesting. A byproduct of this chemistry is also ammonia. So as we reduce carbon, we release ammonia. So 2 of the things that biology needs to fix to make cells is carbon from the environment and ammonia from the environment. So again, is this reaction biological? No. Does it do the things that we want to make cells? Yes, it fixes carbon into sugars and it fixes nitrogen into ammonia. And does it solve all our problems? No. Right? It's again a polymerization process. We're going to make our C2 carbon sugar, our C3 carbon sugar. They're going to be made in the same place at the same time by the same process. And the C3 one's going to isomerize to give us a ketone that we don't want to react with instead of the aldehyde. So there's still more problems, but we're still learning. So, and this is all more complicated, right? If you look at the actual paper, I go back one, this paper, this is a subset of the reactions they study. In here they make components that could make amino acids, they make things that could make sugars, go on to nucleotides, they make types of compounds that could become lipids. So again, we still have this, maybe we're getting closer, but we still have these problems of complexity in a chemical sense. We're building lots of molecules, we need them to operate together in one system, but we need to handle them separately. So there's still more to learn. This is not an answer, this is not the end, this is just another step. And so I have a video, I'm going to see if you can hear it. This video, oh, where's the sound? This video is specifically about ribozymes. It's made by the Shostak lab. They're thinking about complexity in terms of ribozymes, and they're thinking, why do I need a lipid? And this is part of their systems methodology for thinking about why am I going to make membranes and genetic material at the same time. I think it's informative. I think it's worth listening to. I hope you can hear it. That is not what I want to do. Why I wanted to show you that is just, it's a really nice concept. It's a really interesting idea that if we bring these things together, whichever angle we're taking on it, we lose something from the total system if we separate these parts. So for ribozymes, if you love RNA, then you need to separate out this RNA for its function to be expressed in a useful manner. And this is why compartmentalization would be useful. But you saw it, right? We had this messy mixture of stuff. One of these things was useful. And what we want to do is pluck that one out. You saw the magic, right? There was this twinkling. And it appeared inside the vesicle. So what this video tells us is that we know what we want to do. We want to put the bits that are useful in a specific place such that they can express the function that is then valuable moving forwards in terms of making something that can operate like a cell. But we have no clue about how to do this. The way we're going to present it is just the twinkling of the change. Flashes and disappears. It goes from where it's amongst so many things it can't replicate itself, it can't function in the way that we want it to, that it's then isolated. So then that becomes a really interesting question. How do you do that? If you evolve RNAs, it's easy, right? You just put them down a Celex column. You just select them out. You use your methods, your tools in a biology lab to pull out the ones you want. You tag it with biotin, you pull it out, and you go forwards with nice clean material. There is no biotin. Well, there is no biotin extraction at the origin of life. We need to understand more about how we get to the point where we have functionally clean material. So we need to define mechanisms and methods that we can do this. So how does this chemistry self-sort? How do we organize these molecules in such a way that they self-organize, self-assemble, I called it before, but perhaps self-disassemble is also important to us here. How do we segregate and separate? So by knowing that there's something wrong with our chemistry that we can't achieve without me spiking the reaction with the right material at the right time, we can ask new chemical questions. How do I achieve this thing that I do in a chemical method that is hands-free? So separating these two substrates that we wanted to use. We defined some chemistry, meaningful or not, with relevance to the origin of life. Doesn't matter for defining our new experiments, but we can define something we want to achieve, right? We can set up a chemical study that we can look at, something we can meaningfully address in an empirical reaction. How do we separate these two? Now, we are in the dark. How do we define how we're going to go about this? So I want to run you through the thought process, more or less exactly, I think, how we thought about this process, what we thought about, how we looked for a method to do this, and stumbled into this partly by accident and partly by recognizing an accident was useful for something else. But so where it comes from is thinking about nucleotides, one system, and protein type molecules, amino acids, in relationship to each other. So I said many times, we want to think about this holistically. Once we've defined something over here, we have to think, where does that fit in the context of the rest of our chemistry? So we have a nucleotide synthesis. We like the look of this. It has lots of properties that advances from where we were before this. So what more can we learn from now knowing this? The best way to learn from this is to learn from our mistakes. And our mistake, as it might be, is that we have a difficulty of separation between these compounds that appear to be synthesized through the same process and are likely to be made together in the same place. But we want to use them separately. We could separate these with chromatography, right, in the lab. That would work, but it's not a realistic solution to the origin of life. So where do we get new information from? Where we looked for new information on this system is this reaction. So nucleotide synthesis sits over here. This is our prebiotic version of nucleotide synthesis. This is a fairly well accepted prebiotic version of amino acid synthesis. We take an aldehyde, which already bears some relationship to what we're doing over here. The precursors are the same oxidation level, same types of compounds. If you remember the really complicated slide that I glossed over really quickly from the Sutherland lab where they made these sugars and I put it into context, they also made lots of these aldehydes at the same time. Okay. So the chemistry to make both of these sets of molecules looks similar. The chemistry to do this reaction and this reaction, therefore, we would hope are compatible. Right? You could run this reaction and this reaction simultaneously in the same place to give DNA, RNA type precursors and protein type precursors. Is that then true? So we have to look at how these reactions operate and operate together. Well, there's lots of things we could discuss here, but the first thing that jumped out at us when we're looking at this system, we sat down and looked at this, and it jumped out at us because we already knew this, right? But we recognized, that this compound over here is one of these compounds over here. Right, so the first sugar that we use, the C2 sugar, glycolaldehyde, is one of the aldehyde precursors of a protogenic amino acid. It's the precursor of serine through this chemistry. So this, as we analyze this together and we think about it in relationship to each other, we think about the reactions running together, just a thought experiment, places glycolaldehyde as a node. It places it as a node between protein synthesis and nucleic acid synthesis. And we took this to be important. We took this to be a clue that maybe we're right. Maybe the chemistry that we use over here is related to the chemistry we use over here, and they all are expressing the same types of products from the same types of intermediates. In fact, exactly the same, an identical intermediate. It's just reacting with nitrogen, 2 nitrogens and 1 carbon that are in different oxidation states. So ammonium cyanide goes this way to stoichiochemistry, and cyanamide goes this way towards nucleic acid chemistry. So that clue is this compound here, which is the first intermediate en route to nucleotides as we present, and then has to react with another aldehyde, 3-carbon aldehyde, to make this compound. This compound here will not react with any of these other amino acid substrates. Try it in your lab yourself if you want. We tried to get lots of these to do this just out of interest, but things like acetaldehyde are not reactive enough to undergo carbon-carbon bond formation. Formaldehyde, which gives us glycine, does not react with this substrate. So this reaction is specific to it being an electron withdrawn aldehyde, such as a sugar, and doesn't operate for the less electron withdrawn amino acids that go through into aldehydes, that go into amino acid synthesis. So that's interesting. Glyceraldehyde, I haven't drawn this out here, the substrate that you'd want to take through this pathway here, will not do Strecker chemistry. So to do a Strecker reaction, we have to add cyanide and ammonia to glyceraldehyde. But if you take glyceraldehyde and ammonium cyanide, because there's an extra alcohol out here, it cyclizes before the ammonia gets in, and you can't get through with longer sugars. This is the only sugar that will go through Strecker chemistry unless you protect. Put silyl groups, do all the tricks that organic chemists use, but free sugars won't go through Strecker chemistry. They do Kiliani-Fischer chemistry instead. It's known in the literature. So more evidence that we can run these types of reactions at the same time without having the expression of other products that we don't want to see come through to our final set of products that we want to reflect, we want to look like biological compounds. So then we ask ourselves, there is one outlier to this which will participate like glycolaldehyde in both of these reaction pathways. And this is beta-mercaptoacetaldehyde. If I swap oxygen for sulfur, then I have the Strecker precursor of cysteine rather than serine, so that would have a sulfur here as the X instead of a oxygen. So we would expect this, we would hope this was in our product mixture, we would hope that it would be going through the chemistry on this route. So then we have to ask, what will it do on this route? It's going to still react. If I replace the oxygen with a sulfur, I'll get the same chemistry and I'll get a thiazole. Okay? So this analysis is the process we went through. We thought about this in great depth. We thought about all the things we might and might not get. We drew them all out. We considered them all. And then we got to the point where we had this. This doesn't look like an amino acid. It doesn't look like a nucleotide. It's going to be in there. So then we ask ourselves, what does this do? How does this influence the system? How is this going to change the chemistry? That we have this byproduct, this thing that's part amino acid, cysteine precursor, and part nucleic acid. So it's made from two things that are designed, or we hoped were destined to make amino acid and nucleotide. They came together and made something that was neither. It didn't look like it was en route. We ummed and ahed about whether this might influence DNA synthesis. This was our first idea. It didn't. But what we did find it did, and this took a lot of playing with mixtures to find this happened, is that it causes the segregation and separation of complex mixtures in a way that is incredibly favourable to the things that I just described to you, the things that we want to do. So what this is, and I don't want you to interpret this, this is an NMR spectrum. We do lots of NMR spectrum in my lab. It's an NMR spectrum of every sugar that we had in our lab. They're all mixed together. I think they're 50 millimolar, I can't remember exactly the concentration of this. Together, the same concentration, just mixed together as a mixture, as a model formose type mixture if you want. But just as a mixture of sugars, if I have this mixture of sugars, how do they behave? If I take this mixture and then I either make in situ through the reaction was on the last page, or I just drop this compound straight in, what you find is that this molecule, this byproduct, this hybrid of amino acid nucleotide chemistry that we identified through our Gedanken experiment of how this chemistry might go wrong, gives us product extraction. So I take this mixture, I leave it in the presence of this compound, neutral pH, I think it was buffered with phosphate. Can't be certain, I don't think it needed to be, but I think this reaction was, and what you see is one precipitate. You get one compound, which is cleanly extracted from this complex mixture. And that compound is shown here, and it's made from two of these and one of these. So here is the red compound, which is here in the mixture, and it's extracted out cleanly, alongside 2 of these aromatics, and it crystallizes from this mixture. It's separated cleanly. If you look at the supernatant, so this is what's in the solid, dissolved here in DMSO actually in this reaction, so we can see what's in the solid. Here's the crystal structure of the solid. But if you look at what's left in solution, here's where the red compound is. It's kind of almost still there. You can kind of see it if you know where it should be, but it's gone. It's extracted, it's clean, it's removed. So we took a mixture of a great number of compounds and we extracted from this mixture the one compound that we wanted, the first compound we want to use, and left all of the others behind. If we let this reaction keep running, the second compound comes out. But now notice there's something slightly different here. So we can start this reaction in one of 2 ways. Either we put in the C3 sugar that we want to react with, glyceraldehyde, which looks like this, but with carbonyl here instead of the 2 nitrogens. Or we put in dihydroxyacetone, which I showed you before, which is the isomer of this compound, where we have a ketone in the middle and 2 hydroxymethyls on the side. These 2 equilibrate in solution. They prefer to be dihydroxyacetone. This is also a problem for metabolism. If I get as far as metabolism in this talk, I'm going slowly, I might not get that far, but it's a problem we have to solve there. 2, but if we take this compound, you'll notice here is the compound shown in green, it's dihydroxyacetone. If we start with the 3-carbon sugar glyceraldehyde at the start of the reaction, by the time this one's crystallized out, all of this one has gone and has become dihydroxyacetone. This looks like a nightmare for what we want to do. Right? This looks like we took the C3 sugar we wanted and we lost it. If we do reactions with this dihydroxyacetone, this ketone compound, we're going to get isomers of RNA, not RNA. There'll be branched sugars instead of sugars. The wrong thing. But if you look over time, so over a couple of hours, we get complete sequestration of the first C2 sugar. Then over a few days, weeks, you start to see precipitation of a second compound, and this is the last compound we see precipitate. We left these a very long time. We still never, I think they're still there. Nothing else precipitates or crystallizes. Nice big excess of this. There's plenty to crystallize more, but it doesn't happen. If you notice, in the starting material, I have 1 singlet where all of my protons are the same because it's symmetric. It's dihydroxyacetone. In the product, I've got 1, 2, 3. 3 environments. It is now the aldehyde. So the process of precipitation here not only separated the C2 and C3 carbons, it did in a way that we didn't really expect at the start of the experiment. We didn't expect the isomerization to go so fast. We actually thought this was bad when we were running the experiment. We lost the thing we wanted. But when we kept the experiment running, what we found is that this isomerization let this happen. So because our 3-carbon sugar isomerized to dihydroxyacetone, it changed its reactivity. It inhibited the rate at which it was extracted from the solution. Glyceraldehyde, if it isomerizes, gives back glyceraldehyde. Whatever we do to it, it's glyceraldehyde. It's symmetric C2 carbon sugar. So if I isomerize it, I get exactly the same compound. So it comes out super fast, super rapid. The 3-carbon sugar, because it can form a ketose, is then separated out. Its reactivity is different. It's changed. Not because of something we did, because of the difference in 2 and 3 carbon sugars. And then we can selectively sequester C2 sugar and C3 sugar and separate them. Okay? We've not done this, but you could imagine, we always like this idea, you could imagine that this separation happens in flow. If that temporal separation, extract one, then the other, happens in flow, I get spatial separation. This one comes out first, keeps flowing, the second one comes out second. I have now a mechanism, a reason, something based in chemistry, in physics, that allows me to propose and demonstrate the separation of these 2 compounds, and specifically these 2 compounds, not all of these other sugars, and specifically in the order we want them. This feels important. Maybe it's not, but it fits what we want. Obviously, we didn't extract the compounds that we use. So now we need to ask new questions. So we found we can separate these two as these other compounds. They're sequestered. They're in some other form. Some people have suggested to us we should call these organic minerals, right? I don't know if I like that term. They're crystals. They're crystals. But those crystals are separated, but they're not the initial compound. Do they still behave like the initial compound? That's what's important to us here. Not are they the compound? Do they have the same reactivity as the initial compound? And the answer is yes. If we take our first solids, and we don't dissolve them now in DMSO obviously, we just take the solid and add cyanamide to this first solid, we get our first intermediate of nucleotide synthesis completely cleanly. Extracted from the solid, liberating back our initial thiazole compound, which could in theory be catalytic and go back round somehow and do this process again. But then you've got to think about the chemical engineering of actually getting it back round into the start of the reaction. But we'll shelve that. Don't worry about that. What we get is something that is our nucleotide precursor, and we get it cleanly mixed with this compound. If we then take that mixture, and this is important now, we're not now taking clean compound, we're taking the mixture that we generate here of two compounds, and we add this into this compound here, which is again still a solid. So we take the solution we got from our first solid and we add it onto our second solid. And what we find is we get a mixture of amino oxazolines. Right? Our 5 carbon sugars, which look like nucleotide precursors, they have this 5 carbon sugar, they're not branched, they're linear, we've got the nitrogen where we want it, the chemistry is still behaving the same way. In solution now, the only difference is we get the isomers of our nucleotide precursors, 4 of them here, and residual 2-aminothiazole. So it goes from being the compound that extracts our desired sugars from the mixture in the order we want them to a spectator in these reactions. It's just left behind. It's not incorporated into the product. It doesn't make new bonds. It's pushed out of the compounds we're looking at, we're interested in, by the second reaction that takes place. And we get this mixture. If we leave that mixture, this mixture again crystallizes, the same type of process. So these 4 amino oxazolines crystallize to give us 1 amino oxazoline, which is riboamino oxazoline. So we start from a really, really complex mixture, which does actually contain ribose, but we left that ribose behind. Because getting that ribose out, selecting ribose away from lyxose and xylose and galactose, how would I do that? How do I differentiate ribose from all these other sugars? That's a really hard challenge, right? What's the difference? There's just a stereochemistry different. I've got to come up with some complex binding strategy that recognizes stereochemistry. That's tough. This process of pulling out the simpler, smaller sugars is easy because their chemistry is different because they're smaller. Because they can't form cyclic structures in the way the bigger structures can. They aren't just stereochemically different from each other, they're physically and chemically, in terms of their reactivity, differentiatable. So we have mechanisms that are realistic that we can apply to these compounds. And then, fortuitous as it is, riboamino oxazoline is not only the major compound we synthesize here, if you go back, it's this one here, arabino is this one. It is also the compound which is most crystalline, by far and away the most crystalline compound out of all of these amino oxazolines, and it crystallizes out pure. So we start from really complicated. We pull out different compounds and eventually end up with riboamino oxazoline. If I took that nucleophile here, cyanamide, and I reacted it with ribose, God knows where ribose is in there, I couldn't pick it out for you. It's one of the compounds in here. I would have got this compound, but in amongst 20, 30 more other products. How do I pull that out? How do I separate it? I've got to keep asking myself, how do I move forward in a way that's sufficiently like biology that I'm interested in these reactions? And aside, I'm not going to go into the details of this, riboamino oxazoline also crystallizes, looking for serendipity, in a way that amplifies enantiomeric excess. So if there is any EE imbalance in these, it won't happen if it's completely racemic, but if there's an EE imbalance in this substrate, it will amplify that excess as it crystallizes, so gives extra benefits, things that feel important, moving from simple chemistry of compounds that in the beginning, dihydroxyacetone and glycolaldehyde and cyanamide were completely achiral, to the point where we have homochiral biological molecules. So that is beautiful, and there's nice mechanisms to get from here through to nucleotides. That's where we started. But now we've addressed that question. So we got into this chemistry by thinking about sugars. How do I separate and select sugars from complex mixtures? What do I do to get to that point? The way we came up with a mechanism to test something was to think about amino acid chemistry. So to verify that this is actually useful, we thought, well, we should take the same strategy and apply it back to amino acids. How does this thing that was useful for nucleotide synthesis relate to amino acid synthesis? How does it fit holistically into this whole set of reactions? So I have this separation, this segregation that's going to work for sugars. What does it do to the complex mixture that I might have that's en route to nucleotides? And how can I think about that intermediate as a common intermediate that's going to affect both of these pathways simultaneously, productively, and in a way that funnels us towards what looks like it's going to become life? So here's the same reaction. We've looked at this a couple of times. If I take certain aldehydes, some of which were in the synthesis I showed you before from the Sutherland lab, we can take them through to make amino acids. So here's glycolaldehyde, makes serine. We've discussed this one already today as well. Here's formaldehyde, makes glycine. But there are only 20 compounds on this side that go through to the 20 amino acids that we have in biology. Why would we select them when we know, and we look at things like Miller chemistry? I showed you some Miller chemistry before. Someone came to ask me about meteorite chemistry in the break. We know from both of these chemistries, abiotic reactions can give us lots of substrates, lots of products, lots of different types of carbonyls that could potentially become lots of different types of amino acids. So all of these are chosen by us specifically because they kind of look a little bit like each other. They've got similar functional groups, and you can find them all in meteorites, Miller type chemistry. These are the types of compounds that quite high energy abiotic chemistry can give us. But they're two distinct classes because I define them as such, because these are expressed in genetics and transcribed into translated into proteins through the genetic code. I'm not talking about how to encode these, but these are the ones that I want to encode somewhere downstream when I unite these two things together, amino acids and nucleotides. These ones are products of the organic chemistry that might be abiotic, but are not assigned to the code. Why not? They could be pretty useful in making proteins twist, change shapes. Certainly lots of these have been inserted chemically into peptides to make functional, useful protein molecules. Why not use these? Is it really selection? Are we again going to give this problem to biology, just say we can't solve it? Biology, you'll have to select, you'll have to choose these, come up with some mechanism to do this? Or can chemistry do this anyway without that selective input? So we started here, and I start here again, and it was really the chemistry we'd already done that prompted us to do this, because this compound we've seen as well. Remember this guy? This is dihydroxyacetone. This is the compound that became glyceraldehyde, that became the sugar, the symmetric C3 sugar that we didn't extract. So we already know this guy doesn't extract. Right? It will preferentially rearrange to its aldehyde and then come out of solution. So if this one doesn't extract, why would acetone extract? Why would monohydroxyacetone extract? Why would any of these other ketones extract? We don't expect them to. If they don't extract and we don't precipitate them, then when we separate, we're going to wash away the red ones and leave behind the blue ones. If we're doing chemistry on the substrate that precipitates, this gives us a mechanism to separate protogenic amino acids from these alpha-alpha disubstituted non-protogenic amino acids. So here's the same kind of reaction. Again, NMRs. I don't want you to be able to interpret the NMRs. They're complicated. There's 8 compounds in each of these. They're color coordinated. So all the ketones here are in red. So anything marked in red is a ketone. Black is a bit of methanol that comes with the formaldehyde that goes into this reaction. It doesn't matter. It's just going to be left behind there supernatant. And all the blue peaks are derived from our aldehydes. So we start with this mixture again, and we do the same process. We make in situ or deliver our same hybrid compound that was made partly from a cysteine precursor and partly from a nucleic acid precursor, and we see the same thing happen. We get solids formed. The supernatant only contains ketones. All the aldehydes gone completely. In the solids, what we have are the aldehydes extracted as their aminals. We treat them aminals now with cyanide. This liberates the aldehydes, releases the amine that was bound up in the solid as part of one molecule with these aldehydes, which then just sits as a spectator. We get the cyanohydrins of these compounds, no amine formation between here. I found this remarkable, crazy when we did it, that it didn't. We need to make an imine to form this aminal, but the imine doesn't form in the cyanide chemistry, and we just get the alcohol in this compound. And then we add ammonia, and the amine all of a sudden can form with cyanide, and we go through to amino acids that aren't derivatized with this other amine that is now a spectator, but had previously changed the composition of our aldehyde ketone mixture, allowed us to extract the aldehydes, which are precursors of protogenic amino acids. Too many P's altogether to pronounce. So it allowed us to do the same types of chemistry by considering the outputs of our Gedanken experiment, our thought experiment about how the two systems would work together. We came up with a solution for sugars. The same solution applies, the same solution is productive, the same solution works for the amino acid precursors to clean them up too, which was a happy day. So this is a lot about how we make progress. This is our thought processes. I'm trying to show you how we make leaps of the imagination, what we'll do next, how we'll make progress, how we take what we loosely define as a scenario where something's happening chemically and apply it holistically to the whole system to define and find new things. So the next reconsideration we made was to think about, well, these sugars, if we think about them in the context of where they came from, we're going to accept that we get them from this reduction chemistry that maybe is driven by hydrogen cyanide. We then need and hydrogen sulfide. We then need to consider these sulfides in the context further of nucleotide chemistry. So what you saw previously was a sulfur compound. There's no sulfur in nucleic acids, right? That helped us to define the route through which we would take from complex mixtures to get through to simple nucleic acid precursors. What else can sulfur do in this system? How else is sulfur going to influence the chemistry of nucleic acids? What are we missing by ignoring sulfur? We think sulfur should be there. How will it influence the synthesis of nucleotides, even though the final product doesn't have sulfur in it? What does this mean for the chemistry? We wouldn't, if we just considered nucleotides, put sulfur in the reactions. There's no sulfur in RNA. There's no sulfur in DNA. Why would we introduce sulfur to our reactions? That's only surely going to give us byproducts we don't want. Is that true? That's an assumption I just made. I can test it, right? I can take some of the intermediates, the types of compounds we make, and I can evaluate them in the context of, is it geochemical? Probably not at this point, but in the context of a holistic system. How does sulfur influence nucleotide synthesis? So the nucleotide synthesis I showed you, and I'm not going to show you the full synthesis of this compound because it's a lot of chemistry and you guys don't want to see more chemistry, but went to cytidine via an anhydronucleotide. We can propose similar mechanisms to go through to compounds that look like they're en route to purines that build anhydropurine compounds. Okay, so this is such a compound. So this is 2,8-anhydro 8-oxoadenosine, which we can build through similar types of chemistry to what we did with the pyrimidines. How does sulfur influence this chemistry? Well, what does it do? So here's the clean compound, and we add to this compound hydrogen sulfide. It adds. It specifically, cleanly, near quantitatively, there's a little bit of isomerization here. I can explain what this compound is if anyone cares. But it adds to this position here. So it opens this up in a really interesting way. This position is impossible to hydrolyze. We have tried our very hardest under all conditions you can imagine. To hydrolyze this bond, it will not. But if you add hydrogen sulfide, it fairly cleanly, very rapidly adds to this position here, and you get this thionucleobase. Doesn't look like a nucleotide at this point. It has this extra sulfur compound. But wait, we added sulfur, but we didn't add the other thing yet, right? We need sulfur and light. Okay, so to eject an electron from sulfide so that we can reduce hydrogen cyanide, we added hydrogen sulfide and light at the same time. If we then add light, or we irradiate at the same time, it doesn't look as pretty if you just go from here to here. You can't see what's happened. But if we make this compound is then photoactive. This compound, anywhere between 340 nanometers and down to about 200, can't go much below that because we're in water, nanometers, reduces. So the sulfur falls off, and we quantitatively go from this thionucleotide to the actual nucleotide that we find, well, no, the stereochemistry is wrong here, but the actual nuclear base that we find in biology. Okay, so by adding back in the things that we think should be there, just because they were in the system, we can discover new chemistry that is actually productive, that we wouldn't necessarily have looked for if we had limited ourselves just to RNA chemistry and the precursors of molecules that look like they will be built into RNA. So again, sulfur's being added in the same way as it was in the amino, and then it's being removed in a traceless way. It comes in, it goes out. With reference to these two compounds, you wouldn't know it was ever there. But the product of adding it and taking it out is that we go from an oxidized nuclear base to the natural nuclear base that we find in biology. And we can then stitch this into a larger system, all the same types of reactions, slightly different substrates, sulfur again playing a role in how we build a nuclear base, but in a sense that is traceless. So we're introducing sulfur in this chemistry, even though it's not expressed in the final products, because we expect it to be there, because we want to interrogate how the chemistry will operate in a holistic sense. So this is not the same thing as just taking all the compounds you could ever get, throwing them in a pot and seeing what they do, but it's layering complexity in a way that makes sense, in a way that is holistic, in a way that we can interrogate through reasonable reactions and analyses. So why we found this particularly interesting, or let us get through to the kind of products we wanted, but what really piqued our interest with these compounds was that we did this for lots of variants of this. We did A's, we did I's, various others, but when we did I, it was odd, right? Odd that when you do the same chemistry with A and I fails. So here's a mixture of thio-A and thio-I. We irradiate these two, and what we find is a really good yield of A, but a very poor yield of I. So completely serendipitous. We don't expect this. It doesn't look like these two are going to behave any differently. They look really similar, right? Replace a nitrogen with an oxygen. But through this process, it gives us a mechanism that delivers lots of A and not very much I, which again sounds biological, right? This is in biology. This one is in biology just about. It's not in Watson-Crick base pairing. You find it maybe in some tRNAs, not all tRNAs. But it has that feel of we're going towards the compounds in the kinds of mixtures that we want to build biological structures. I can fully explain to you why this works, how this works, through the mechanisms of this reaction. I don't want to do that because you're not all chemists. But by looking at it, by understanding it, we can know why I doesn't come through, why A does come through, why G also gets through really well, and start to understand what else we might do with this chemistry. Why else could it be useful? So by getting a complete mechanistic understanding, we can then build new questions, new methods to think about why this works. So the final thing I want to talk about, which I didn't know I was going to get to, but I have, is just a little bit about thoughts on metabolism. Is more to provoke thought than to suggest to you that you should think this is the way metabolism started. But again, taking this holistic approach, what can we learn from the things that we already know? What can we do with those compounds? What kind of reactivity we can get? Why would certain molecules that accrue energy from the environment be expressed in these systems? What is it about these systems? And this becomes difficult for a chemist. Right? This becomes difficult for a chemist because metabolism is so many reactions. When I was just considering RNA and peptides, I can handle that, right? They're the types of numbers I can deal with. 4 and 20, that's all right. Stitch them together. I want to be selective about that. A map of E. coli, I think this is metabolism, is so complicated. There are so many reactions. There's so many things going on here. To address this realistically, how do I start? Where do I start? How do I infer some importance to any one part of this? And I probably know what you're all thinking already because you've been told to think it, right? There it is. TCA cycle, that's really important, right? That's at the heart of metabolism. That must be where metabolism starts because all of these molecules meet and join in the middle and run around this cycle. Could be autocatalytic. Could be true. Is it? I don't know. Should we go there? It's not where we went. I'll show you where we went, and I'll show you how maybe I thought we shouldn't go here. Well, one reason we didn't go here is other people had gone there, and we didn't want to follow what other people had done. But another reason was in thinking about networks. This, as someone from the UK, struck me straight away. If I take a complex network that functions, that works, and I want to strip away from it parts, I must be careful. So this is a map of the British rail system in the 1950s. In the 1960s, someone used to work for ICI, a chemical company, never put chemists in charge of networks, but hey, was asked to analyze this network. How do I make it better? How do I make it more efficient? Which parts are most important? And he highlighted them in black. Okay, so there are lots more networks in here as well. These are the main tributaries. And he said, these are the important pathways. Oh, look, there's this little cyclic bit in the middle. All of everything flows around here. Everything comes in and out of here. This is what's important. If we strip away all the other bits and we just leave behind the black bits, then the rail network will be better. These black lines make loads of money, right? They're really highly productive. The other green lines, they are not making any money at all. Actually, we need to support them with money from the black lines to make sure that they operate. So just strip away the green lines, right? We did strip away the green lines. Well, not all these green ones, but lots of the bits in the middle. We took away half of our rail network between the 1960s and the end of the 1970s, and now none of it's productive, none of it's useful, because all of these green tributaries made the black lines useful. They brought in the passengers, they brought in the products that ran down the black lines. If we try and strip away what we feel to be peripheral, often we can ruin the whole. Okay, a little bit of poor management on networks maybe can be thought about in the context of this. Can we realistically strip away E. coli metabolism to just leave behind the things that look like they're most important? Is that a fair thing to do? Is that a useful thing to do? Does that make sense in the context of networks, realistically? I don't know. Maybe it does, maybe it doesn't. Obviously, this is analogy. We're told not to use analogies. And I'm using this analogy not in a way to say don't do this, just to be careful, just to think. Also thinking about what comes first. So here's London. All of our network comes and seems to emanate out from London now. Well, everything's London-centric in the rail network. But here's the network in the 1840s. doesn't look at all like it. In fact, there's nothing central about London because all the people who lived in London were scared of the railways. They thought they'd shake and vibrate their houses down, so there were no railway lines that crossed London. If you've ever tried to cross London by train, you'll find it's a horrible process because none of the railway lines come to the middle. They're all peripheral around the outside, which is why there's a ring of network around here. Whereas the first line's up here in Darlington. That's crazy, right? You've never even heard of Darlington, but that's where it was. And then the next one was over here in Manchester to Liverpool. So these lines weren't assembled in a way that seems coherent and important into how the network works at the end, at their beginnings. Right? There is context to why this network sprung up in the ways it did, in what was going on at the time, where the mills were, where the coal fields were. It wasn't about moving commuters around. So at the start of the rail network, it had a really different function than it did and it does today, right? Because our economy is completely different. So when we analyze a network, we have to analyze it in situ, in my opinion. But having said that, I'm going to not take my advice and I'm going to strip a part out. So there we go. But I'm going to choose a different part. Why am I going to choose a different part? Because I think it's interesting. Because I love this compound. This is the highest energy compound in biology, highest energy phosphate, in biology, phosphoenol pyruvate. That makes it a challenge to me as a chemist. Can I make it? Is it stable? What chemistry gives this? How do I set up this high energy compound that can then fall down in context of metabolism? This is really old, and I also love how interconnected this network is here. I just told you not to do this, but I'm going to do it anyway, because I can define experiments. I'm not saying don't do it yourself. I'm saying just take it with a pinch of salt. Can you realistically strip this network out? Is it still useful? I'm going to show you chemistry now that looks a little bit like metabolism, but what's it doing in reality? Until I put it into a cell, until I use the products of it, what have I got? I've got chemical reactions, not metabolism. So just a warning. So the first thing that we did to set up this process, so what we wanted to interrogate is how this works. Do I need enzymes to make this process work? From going from simple C3 sugars down through to energy molecules like phosphoenol pyruvate, acetyl-CoA. So why we chose here wasn't just completely serendipitous. It wasn't just that it sits here. It was we'd already been playing with these molecules, right? These are the sugars. These are the 3 carbon sugars that we extracted out. So it's a natural progression to move on from the chemistry we were on to ask, can we keep going further? So there's history in terms of how we came to this experiment, as well as just analyzing the energy in phosphoenol pyruvate and how modern metabolism looks and the interconnected nature of these 3 nodes in metabolism here. But then we have to find, again, organic chemistry that lets us interrogate this process. What are we going to do? What reactions are we going to perform? Is it meaningful just to throw sugars in solution and ask what products we get out? Possibly, but it's not how we operate in our lab. We define specific chemical reactions we can control. And this was one. This was a literature reaction that allowed specific phosphorylation of sugars in solution. And we exploited this reaction to quantitatively phosphorylate 2 carbon sugars and 3 carbon sugars. But they quantitatively phosphorylate alpha to the aldehyde. And this was in the literature, but no one had ever done anything with it. No one had ever shown it was useful. No one ever showed what happened. And we got interested in it because if you take both of these compounds, both nucleotide precursors, if you do this kind of reaction, you block nucleotide synthesis. I need this oxygen. I need this oxygen to synthesize nucleotides. Without them, no chemistry. So what happens instead? This compound eliminates straight away. We just warm it up and it eliminates to give a phosphoenol, but not now pyruvate, pyruvaldehyde. Really easily. If I can eliminate this, I can deprotonate here, which means I can deprotonate here. And if I add in, as well as these 2 compounds, formaldehyde, this C2 carbon sugar gets dragged through this process as well. So if I take all 3 of the simplest aldehyde sugars, react them to phosphorylate them on that alpha position, it will all end up down here as this substrate that starts to look like biology's highest energy phosphate and starts to look like it's starting to mirror, but not be, the same type of chemical pathway. Not the same compounds in biology, versions of them, but they're different. And then we can start to manipulate this chemistry further. So if I had cyanide here, I can make the first precursor of the first component on triose glycolysis. Ammonia goes in, I get phosphoserine, that's also important. If I oxidize these, then I make the first, second, third, fourth, keep going down this process. So what did we learn? Well, this isn't biological. I can show you how the process worked. I can show you ways to make it selective, to not do certain oxidations and to do other oxidations, but that's not what I want you to take from this. What I want you to take from this is the reconsideration that we made. So we looked at this pathway and we asked ourselves, how do I make this chemically simpler? How does this work without an enzyme? And what we ultimately did when we look back at the process is, instead of oxidizing here, like biology does, to make a carboxylate here, which then can't eliminate, we phosphorylate at the aldehyde level so that we can transfer phosphate intramolecularly. The aldehyde, because it's not the acid, activates this position to elimination, and then we oxidize. So all we did, other than doing the chemistry with a different phosphorylating reagent, is swap the order. We oxidize second and eliminate first, whereas biology oxidizes first and then eliminates second. If you oxidize first and eliminate second, you have to force this chemistry through with enzymes. It's incredibly slow, it's really difficult chemistry to do. It stalls, it stops at each stage. If you go aldehyde, eliminate, and then oxidize, so swap the order of those two, the chemistry will just run through. It will take you through to phosphoenol pyruvate. And then that lets us ask lots of interesting questions moving forwards, which I'm not going to answer here because I'm out of time and also don't know the answers. But then we can start asking questions about this running as a network. Why is it that biology runs this the difficult way instead of the easy way? Is it important that biology stalls and stops and switches on and off the flow of material through these metabolic networks? Is it important that it's not just easy to release material through metabolism? Is the real process, the real important thing we should be considering in terms of enzymes, catalysis and metabolism coming together, not speeding things up, making things happen faster, but slowing them down, letting us switch them on and off? Because then that's starting to sound like a real network, a real logic system, where you can actually get products of definition by the type of catalysis you have, based on which reactions you allow to go and which reactions you're allowed to stop. So we need to think beyond. As soon as we start to think about metabolism, we need to think beyond just making stuff. We need to think beyond just speeding stuff up. We need to think about it properly, like a network. Like the British rail system wasn't thought about as a network. How do all the parts communicate? Why is it that these operate in such a way to support cellular evolution? Not as an independent entity, again, as a part of the whole, as a holistic approach. And I won't go over this again. This is just to say that this is triose isomerase, needs a really, really powerful enzyme, TIM enzymes that operate at the limit of diffusion in biology. This is a really, really highly evolved process. This is the same type of reaction that we did with our simple crystallization process, turning dihydroxyacetone into glyceraldehyde. So again, feeling that these fit together, feeling that we're working towards one holistic process. But there's loads more to do, loads for everyone to do, way more than just our lab could possibly consider, to get from the point where we can build these, assemble these, have them operate together, such that they can do this last bit of Darwin's thing, right? That they can start to change of a way that is meaningful, start to really participate in what we would think of as evolutionary experiments. So I'll end there because I'm out of time, a little 1 minute over. But I will thank the people who did this. All of this work was not done by me, obviously. It was done by these people. These people funded us. These people collaborated with us. You may listen to me, which I thank you for too. And I thank the invite for the invite again, because it's a fantastic thing. I thank you. If any questions, I'll answer them.